Welcome back to the module of rod and neurosurgery, and the last session was about spinal neuroanatomy. So this entire module we are trying to cover the spinal surgeries for various experimental spinal procedures. So wherein the objectives will be to study the spinal cord structure or to prove the various centers or various pathways that will be useful for various translational neural engineering work. So last time we saw that we covered extensively about central nervous system, spinal skeletal system and spinal cord vertebrae relation. So just to have a brief recap, we saw how spinal cord forms a very important conduit to convey the information starting from the brain towards the end organs like muscles or glands and heart and various other organs. And then we saw how the spinal skeletal system envelops the spinal cord and protects it, and in order to approach what are the layers that we need to sort of excise or reveal, and more importantly this spinal cord vertebrae relation as to how each spinal cord segment is in relation with the different vertebrae. So with that we have covered the majority of the skeletal system. So today we will deal with the actual spinal cord and what are its vascularity and how to take care of the vascularity before we approach them, and then briefly about the cross-sectional anatomy and the actual spinal cord structure, what it encompasses like gray matter, white matter, what are the circuits that are involved. So these kind of knowledge is pretty important before you begin your experiment, or for that matter if you have your research question ready, how to sort of relate with the knowledge on spinal anatomy, which is very important to have a very good experimental outcome. So with that brief introduction, so I will try to show the layers now as to how in reality how many layers that are needed to dissect out before we reach the actual central core of the spinal canal, that is spinal cord. So on the left hand side I have shown the rat mainly to orient yourself again to what is dorsal and ventral. So what has been exposed here is the glabrous part, that is after shaving the hair you will see this outline of the spinous process, and I hope you all remember what the spinous process is, that is neural process, which is the dorsal most portion of the vertebra. So if you all remember from the last session I showed that each spinal vertebra is around the spinal cord like this. So this is the part that we are trying to expose by making an incision, that is here. So if you all remember the anatomy, that will be the cranium from where the spinal vertebra starts and then it ends in the tailbones, alright. So spinal cord runs right in the center of the spinal canal. So in order to expose you need to go in that direction to expose the spinal cord. So every layer in this slide is from above downwards. That means we will be exposing the skin, then there is a muscular layer, then comes the bone, then the dura mater and arachnoid, then the actual spinal cord, alright. So these are the layers that you will be seeing in each subsequent slide in order to approach the spinal cord. So that is rostral, the rostral is this, caudal is towards the tail. That is how the dissection is going to progress, alright. So that is before making a skin incision. Once you make the skin incision, the first layer is the muscle. As I said, if you all remember the three-dimensional anatomy which I showed last time and the various muscle group that I explained. So obviously the subcutaneous tissue will be along with skin, The subcutaneous tissue fortunately is pretty thin over the dorsum, over the spinous process, but there is this body habitus that we will be discussing when we probably deal with the animal handling and animal selection sessions. You need to remember that if the vertebral body is very prominent and it is seen right on the surface, that you can count these spinal vertebra, that means the rat is pretty emaciated. So, that is the subcutaneous fat is lost, and then you are able to see the spinous process. So, then the rat's nutritional status is very poor and may not withstand the surgery. So, there is a thin layer of subcutaneous fat, maybe here you can see the outline here. Once you reflect the skin, there is a thin layer of subcutaneous fat which usually moves out, and there is various technique to hold it back with multiple retractors that way, which we will be discussing it in the spinal surgery section. So, here what you are seeing is the outline of the spinal vertebra there, and these are erector spinae muscle, which will maintain the posture of the rodent when it moves. Because spinal skeletal system needs stability on both the sides for the purposeful movement of the rodent. So, there are various paraspinal muscles which I showed you last time, which is important to maintain the posture. For example, if this part of the muscle is weak, then this part of the muscle which is active will pull the spine towards its side, and then you will get what is known as scoliosis deformity. Alright. For example, if there is a malformation which is in front, that is ventral, then you will get what is known as kyphosis, which is known as hunchback. Alright. And that is how the spinal skeletal system will bend, and that will call kyphosis. Sideward bending is known as scoliosis. Together it is known as kyphoscoliosis. So, all these are pretty important to remember, because if your experiment involves catheter injection into the epidural space, if you are planning to tap the spinal canal by way of injections, which we will deal with in the spinal surgery topics, then the anatomy of the spinal canal should be normal. Only then you will be able to pass the needle blindly in between the spinous process. That is, in a sagittal plane, this is how it is going to look, alright, and that is the spinal canal, and you are trying to take the CSF sample. I do not know how many of you are familiar with a procedure called lumbar puncture, alright, lumbar puncture or any level CSF tapping. You are trying to take the cerebrospinal fluid from the arachnoid space for various researches, or you are trying to instill drugs into the spinal cord or into the spinal subarachnoid space, then this anatomy is very important. There should not be any bend in the spinal canal like scoliosis or kyphosis, because the anatomy is different and you will not be able to tap the CSF canal. So, that rat should not be selected for the experiment. If your experiment involves such a very delicate procedure which you have to go through this spinous process blindly without any image guidance, then you need to make sure that there are no skeletal deformities, alright. So, that is one thing that you all need to remember at this point of time. So, here what you are seeing is the spinous process. So, that muscle has been split. There is various technique to split the muscle, which again we will discuss it in the spinal surgery section. In the anatomy, what I want you all to appreciate is that once the muscle is cut along the sides of the spinous process, you will see the actual spinous process coming into view, alright. So, that is rostral, this is caudal again. So, the spinous process comes into view, and then if you dissect it a little more further, the technique is that you need to stick to the bone in order to prevent injury to the spinal cord which actually will be partly exposed in between bones. For example, if this is bone and this is another bone, you will see spinal cord somewhere there. So, if you are not careful, if you are using a cautery, Bowie cautery or bipolar or monopolar cautery especially, there will be heat induced damage if you are not careful. So, the idea is to use the blunt dissection, use your dissectors right on top of these bony process and keep pushing the muscle away from the bony structure in order to go deeper. Alright. So, once you do that, this vertebral anatomy will be appreciated. So, what you are seeing here is the T13, that is 13th thoracic vertebra, and this is the spinous process of the L1 vertebra. So, that is very important to keep in mind. So, once these landmarks are exposed, then comes the actual drilling or removal of the lamina, the process called laminectomy. I am sure all of you must be familiar by now as to what the laminectomy is. So, then what you see right underneath is the dura covering the spinal cord. There is a very thin layer of dura here which is covering the spinal cord. So, just to keep your orientation, that was the spinous process which you exposed in the last slide, and then you removed the lower part of the lamina. Then the dura will come into view. So, if you remember sequentially the structure, first is the skin and then the subcutaneous tissue, then the muscle plane will come. Once you dissect the muscle plane, you will see the spinous process. When you deepen it, all these structures of the vertebrae comes into view. Then comes the dura mater covering the spinal cord. So, these are the structures that you need to dissect or remove before approaching the spinal cord. So, it is a pretty demanding microsurgical step with significant learning curve. If you are not careful in any of these steps, the delicate spinal cord will be injured. And moreover, here by mistake it has been shown as artery. Most of the time it is vein that you encounter first. There may be dorsal spinal artery, but most of the time it is dorsal spinal vein what you see here, and that will be injured and the entire cord will get affected. I mean, if it is the contusion model of spinal cord injury, then fine, but then you are supposed to do a controlled contusion, not the injury to the spinal cord while you are doing the surgery. Alright. Even if your experiment involves the contusion of the specific segment, you need to be careful before you actually bring in the impactor and cause the contusion. It should not be that your surgical instrument should cause the injury, because that will be unpredictable. You will not know the depth of injury and it might affect your behavioral outcome of the experiment. So, once the dura mater is exposed, next once you remove the dura and arachnoid, what you see here is the dorsal surface of the spinal cord. So, you are seeing small vessels which we will discuss. So, this is how the spinal cord will be visible, and then what you are seeing along the side, these are the rootlets which we discussed in the previous session, and this is how the spinal cord segments will be named. For example, if here is the T13 cord segment, I mean if this was the T13 vertebra, if you remember from the previous lecture, we need to add at least +4, then this will be what segment? This will be around L3 segment of the spinal cord, because this is under the T13 vertebra, which will house maybe L2, L3 segment of the spinal cord, and this is how the rootlets goes. That will be L4 segment of the spinal cord, that would be L5 segment. So, each rootlet corresponds to one cord segment here, alright. So, that is how, look at the length of the spinal cord, how smaller it is, and as it goes closer to the conus, that is the end of the spinal cord, the segment is becoming shorter and shorter, and that is how each vertebra will house at least 4 to 5 cord segments towards the end of the spinal cord. Alright, so I hope you understood the relation. Each segment of the spinal cord will have the rootlets, which will have dorsal rootlet and ventral rootlet. This is the dorsum. Below this there is another root which goes right underneath, and towards the foramen there is ganglion formation, which is dorsal ganglion, dorsal root ganglion. Then the peripheral nerve starts, which is the interface between the PNS and CNS, which is CNS is the spinal cord and PNS is the nerve, and that is the interface, and that is where usually the tumor arises. Alright. So, it is called Oberstein-Redlich zone, ORZ, wherein there is a stoppage of oligodendrocyte and then the Schwann cells begins. That is the area where a lot of these cell dividing activity happens, and then very different zone. So, that is where in this transit zone is where the schwannomas arises. Alright. Just an added knowledge of applied anatomy at this juncture. Alright. So, I thought of showing you a little bit more details of these, how the dorsal root ganglion is seen, and here at this level, I mean even in the spinal anatomy is pretty complex, alright, because the entire spinal cord is 3-dimensional, the cord is just exiting laterally, that is sideways, whereas you are exposing the spinal canal straight down from the top of the animal, that is skin, subcutaneous muscle and bone. So, when that comes, this is the orientation. For example, if that is the the mouth of the rodent, this is the body with the spinal canal on top, and that will be the tail. Just to give an orientation as to how this skeletal system is aligned, that will be the hip bone of the rodent from where the hind paw comes, that is the hind limb, and that is where the, for example, if this was cervical vertebra, what is been shown is the zoomed view of lumbosacral, that is lumbar vertebra and sacral vertebra. If you remember that 58 vertebra, this is the tail, this particular vertebra will be somewhere here, only this region. So, that has been zoomed in and shown, but just to give you orientation, this is how the rat is lying here. Alright. So, you are approaching from top. So, once you got the orientation, I will just erase this. So, when you expose the spinal canal, as I said, if you continue your exposure sideways, these are the laminas, alright, from the back, and if you take a cross section here, when you cut it perpendicularly to this line, this is how it look like. This is the axial section. If you all remember the plane that we just discussed, this would be coronal and this will be axial, because it is perpendicular to the midline, alright. So, it takes slices like that, and this will be axial. So, that is where the ganglion would be, and that ganglion in coronal orientation, this is where it is. That is the idea of this particular slide, to show you how the dorsal root ganglion appears, and this is a sort of a very important landmark for various researches which involves sensory system. The dorsal root ganglion is the major junction for all the sensory inputs that comes from the peripheral hind limbs or forelimbs, or from peripheries, and for that matter even the trunk send the sensory inputs, all the sensory inputs that goes through the dorsal root ganglion from the peripheral nerve. So, this is the nerve that I was talking about. This is the spinal cord, that is the spinal rootlet, for example dorsal rootlet, then it combines, then it fuses with the ventral rootlet and forms the dorsal root ganglion. That is what has been shown here, alright. Then the nerves, peripheral nerves continues along, for example if there is a limb, it goes and supplies the limb there. That is how the entire peripheral nerve system starts, alright. So, in order to expose this, what they have done is you need to remove the lamina. So, this has been removed. Only after removal of lamina, then you extend a little more on the lateral. All these has been removed to expose the dorsal root ganglion. For example, you compare the bony landmarks on top and below, then you know what has been removed to expose the dorsal root ganglion. We removed one half of the lamina. Alright, that is the outline of the removed lamina there. Once you remove the lamina and the part of the facet, then this nerve will come into view, or the dorsal root ganglion. So, if your surgery, if your experiment involves dorsal root ganglion, this is how the exposure should be done, and these are the layers that you would have to dissect it before exposing those dorsal root ganglion. So, obviously when you are using a bony instruments in this area, we need to be very careful in order to prevent injury to those nerves, neural structure that you are trying to expose and study in first place. And that is why the anatomical orientation, anatomical planes and all the structure are so important to understand before you begin your surgical experiments. So, then now let us go one more layer deeper. This is where we were, alright. We removed the bony structure, and this is when the spinal cord, which is the major neural conduit structure which is coming from the brain with all the information. So, any cut here will leave the person paraplegic. He will not be able to, he or she will not be able to move the limbs, and in rodent both the hind limbs will become paralyzed, alright. So, that is a major injury, and all the spinal cord injury models will target this particular area to induce injury by using what is known as impactor, which we will go through it in detail when we discuss spinal surgery. So, now just to understand, this is the side view, or anatomically lateral view. All the bony structures has been removed to expose spinal cord, and this particular slide is mainly to expose the rootlets that we just discussed. So, this is how the roots would look like. This is the outline of the root, and you can see various arteries coursing around. Those are the vascularity of the root as well as the spinal cord, blood supply, alright. So, these roots exit through what is known as intervertebral foramen, and then it becomes the peripheral nerve here. So, this peripheral nerve is dividing further and supplying the muscle belly. So, this is the muscle belly of the forelimb or the forepaw, and you can see it is supplying the flexors of the elbow, that would be the shoulder region, and one of the major nerve supplying probably the musculocutaneous nerve supplying the biceps brachii muscle. So, we will deal with peripheral nerves and skeletal muscle in a different section, but this is how the peripheral nervous system continues starting from the rootlet, alright. So, I hope you all remember the each and every layer we just discussed. In order to expose the peripheral nerve until the muscle, this is how the neural structures are going to course starting from the brain. So, imagine if there is a motor cortex, a neuron, and this is how the brain is, and then the spinal cord, which we dealt with it now, and then the rootlets comes in, then it goes and supply the muscle which is shown here. So, we finished the brain, we came through the spinal cord, then the rootlets, then comes the nerve through the foramen, then it continues this way to form the peripheral nerve. So, until here we call it as central nervous system, from the rootlets onwards we call it as peripheral nervous system. So, that is what in nutshell the entire nervous system would look like, and that is how the various signals, neural signals will be conveyed from the brain to the end organs. So, But how does the transition from the brain and spinal cord would look like? So just to recapitulate, if you remember, this is the cerebellum, alright. So hidden part of that ventral to it, this is dorsum, ventral, rostrum, caudal. That is the orientation, alright. So when you orient, so this would be the head of the rodent. This is the back portion of the brain, and that would be the lambda, and then the occipital interparietal bone and the occipital bone which is covering. So that has been removed, this entire part has been removed, all the skull bones are removed. This is the otic area, that is the ear canal. This would be the brainstem. Just let me clear it out. This is the cerebellum, that is the brainstem. From the medulla oblongata, you can see that the spinal cord starts and then go down, and you can see this tiny rootlets which are coming out. So these are cervical spinal cord. So this would be C1, C2, C3, C4 and C5 and so forth. So these are cervical roots from the cervical cord. The Latin cervix or cervical refers to neck region. So this would be the neck region of the rodent, and this is how the this is a little bit complex, because you need to deal with the posterior part of the head in order to expose the cervical cord. So it's a little bit if you need to expose up to C4 segment, you need to follow all the stereotactic techniques of head fixation, exposure of the posterior part of the skull, then only you will be able to expose the atlas and the axis. So it is very important to understand the orientation of cervical spinal cord along with the posterior fossa structure, or for that matter cerebellum. So you need to sort of understand what length of the incision that need to be made, because it will be tailor made for your experiment. There are no standard. For example, in brain surgeries, all the incisions were standard, whereas in spinal cord or the spinal canal exposure, you need to understand which is the part of the segment that you need to expose. If you want to expose up until C5, you need to start the incision from there and then go down. Only then you will be able to expose the upper cervical cord. You need to expose the cerebellum and the occipital bone to safeguard those structures in order to expose the upper cervical cord, that is up to C3. Alright, so it is very important to understand the relation with the brain before you actually begin exposure of the upper cervical cord. That was the whole idea of this particular slide. Alright. So now briefly before I wind up about the spinal anatomy, you need to understand the spinal cord arterial supply, which is very important. One is that it might directly deal with your experiment if you are trying to study various stroke models of spinal cord, arterial compromise or arterial malformations, so on and so forth. So if you need to study the arterial anatomy of the spinal cord, and if your experiment deals with it, then you need to know. And of course, when it involves any sort of surgical implants, then you need to know how to safeguard those vessels in order to make sure that the cord is alive. Okay. So as I said, this is the dorsum, this is the ventral, and this is the cross section. Okay. So cross section obviously is taken that way, and then you have the vascularity shown here. So when it is ventral, there is a midline artery known as anterior or ventral spinal artery. Alright. So that is a major vessel which comes in the center. You can see it here in the cross section, which supplies majority of the spinal cord almost up to the anterior 2/3 of it is supplied by the anterior spinal artery. On the dorsal surface, if you all remember, there was roots. There were roots, multiple roots along the sides of the spinal cord, and one of the blood vessel enters and then supplies the spinal cord that way. All right. So, that is how the dorsal part of the spinal cord is taken care of. So, any damage to any of these vessels leads to ischemia or stroke. All right, and that leads to the deficit in those specific areas of the spinal cord. That is very important to remember. So, that is how the spinal cord arterial supply happens, but then if you wonder where it comes from, it all comes from the vertebral artery from top. Maybe I can show here. This is the vertebral artery which is going in the spinal canal. So, one of the artery joins together with the opposite side and then it continues as anterior spinal artery there. Multiple intercostal arteries, that is the arteries between the ribs, they come towards the spinal cord and then supply all these rootlets and also supply the spinal cord substance on the dorsal surface. On the ventral surface, this artery continues as ventral spinal artery. All right. So, it is all little too much to take in at the first time. Maybe if you watch these videos again and again and read up bit more about it, it will be easier to remember. So, because we are dealing with mostly with the spinal surgery, I am trying to cover mostly the applied spinal anatomy and applied cranial anatomy. If you can dig in a little more detail of all these structures, then it would be easier to understand and orient yourself with various anatomical structures. So, now let us look at the spinal cord venous drainage. I hope you all remember, the venous drainage takes the blood away from the organ, arterial supply supplies the oxygen and various nutrients towards the brain or neural structure. What we saw earlier was arterial supply, this is venous drainage, and you can see this is artificial dye injected into the vein. It doesn't really look this blue. This is a blue latex which has been infused after the death of the rodent. You can see that this is the dorsal spinal vein. So, as I said earlier, that is the major vein that is seen there, and it takes away all the blood which has been given by the ventral spinal artery. This is the artery. There is the ventral spinal vein as well, which is very thin. So, what you need to remember is, on the dorsum the vein is very prominent, ventrally the artery is very prominent. You have a big artery running in the midline ventrally, and dorsally you have a big dorsal spinal vein running. That will take all the blood away from the spinal cord and towards the heart. All right. So, it is more or less similar. The orientation, the supply and the drainage is more or less similar, and if you safeguard all those structures during the surgery, not only you will be protecting the artery, but also the vein. All right. So, this is a good picture to give you a three-dimensional orientation as to what is artery and what is vein. On the left hand side, will give you a nice orientation of these structures which we just discussed, and the entire neuroanatomy. I give a lot of importance to this three-dimensional orientation because it is a three-dimensional structure. For that matter, any organ in the human body should be understood with the three-dimensional orientation and its relation with its other structures. More so in the neuroanatomy, because most of the time you will be exposing only part of the nervous system, not the entire nervous system, because the entire nervous system is around it is surrounded by the very strong skeletal system, unlike any other organ. For example, you need to expose the abdominal organ, you need to make one slash in the center, the entire abdominal organ comes into view. With that, the handling of that particular organ becomes a little easier. Whereas in neuroanatomy, you need to plan it, understand, and then do a very small opening to that particular area, deal with it and then quickly close it. And if not, sure about the anatomical aspect, you might miss the structure or you're opening it in the wrong level. That's the biggest problem, and that is why these entire sessions we are dedicating to the anatomical applied anatomical details, so that you will have a good orientation before you even start reading in detail about it. So, if you look at the spinal cord, this is a three-dimensional view, whereas the spinal cord continues that way, alright. So, imagine this is the rat body, alright, this is the mouth and that is the tail. Again, to orient you, this is the dorsum part of the spinal cord. So, that would be dorsum, ventral is ventral, dorsal horn and the ventral horn. So, the inside part of it I will discuss it, but this slide is basically to make you understand how the vascularity is oriented, alright. So, once you understood, this is the posterior spinal vein I was talking to you all about. On the dorsum or on the back of the cord there is a prominent vein running, and in the ventral part there is a huge vessel running in the midline which is ventral, alright. So, this in that surrounds the spinal cord and supply the substance of the spinal cord, okay. And then it takes away all the veins, which is here it is called capillaries. So, when artery becomes smaller and smaller to the end organ, it forms what is known as capillaries. So, these are the bit of anatomical basics which I recommend you all to learn and get familiarized, so that these anatomical details can be understood. So, that would be arterial system, that is the venous system. This is the capillary system, where the most of the oxygenation and nutrients diffuse into the surrounding nervous tissue. So, that is where here. So, this is where the capillary system will be, then the venules will start, and then the vein forms, and then it takes away the blood to the heart, alright. So, that is how, and this is the angiographic. On the right hand side you are seeing the angiographic 3D modeling of the arterial and venous structures, alright. So, you see how the artery divides within the substance of the cord and supplies, and these are the multiple vessels which goes around from the same arterial anterior spinal artery, and this is the posterior spinal arteries which comes from the rootlets and then supplies all around, and the entire cord substance is covered with the arterial arcade. So, if you really want to make any sort of surgical approaches, these arteries should be avoided at any cost. Whatever surgery, whatever experiment that you are planning to do, you need to safeguard and choose a relatively avascular zone. As we discussed in stereotactic brain surgeries, you need to avoid these arteries, then only you can approach the cord substance, alright. So, how does the inside of the spinal cord would look like, and what is it composed of? So, as I said in the beginning, it is a nice little bundle of electrical wires. If I need to give an analogy to the cables, this is how it looked like. You know, there is this, I can say the earthing wire, and then there is a positive, and then there is negative wire. So, there are wires which connects the brain with the end organ, that is taking the impulses from the brain to the muscles, and there are cables or the axonal bundles or the white fiber tracts which carry the impulses from the muscle to the brain, alright. Just like artery and vein, there are motor system and there are sensory system. Similarly, there are motor tracts and there are sensory tracts. Tracts means fiber bundle, alright. Tracts refers to fiber bundle, which are nothing but white fibers. White fibers are formed by multiple axons, okay. If this is the nerve bundle, then there are multiple smaller bundles within it. So, that is exactly the reason why I said it is like a fused cable wires which has multiple small white fibers which are formed by the axons at cellular level. which is seen nicely here, where this is the grey matter. I hope you know the basics, that the entire nervous system is formed by grey matter, which will have the neurons, and then the white matter, which will have the axons. Just to give a brief recap about the basic structure, structural unit of the nervous system is the axon, which will have dendrite, a neuron which will have dendrite, and this is the axon, and then it forms the synapse with the dendrite of the other neuron, and then it continues as axon and dendrites and so on and so forth. So, if you see, there are multiple neurons within this grey matter, which will house the cell bodies. And you can see nicely those axons coming out of those multiple neural cells, and this is how the rootlets forms. So, rootlets basically has thousands and 10,000 and millions of these axonal bundles to fuse to form the rootlet, and then the rootlet will obviously will form the dorsal root ganglion, then it continues as peripheral nerve, which we just saw it in the previous sessions. So, this is how it is. This is grey matter and this is white matter. So, that is in the form of H. Maybe we will have it, yeah. Here you can see this is the actual outline of the grey matter, alright. So, you can see that it is in the form of letter H, and here what you have what known as dorsal horn and ventral horn, and that is the central canal, which will have spinal fluid inside it, which is continuous. This is in continuity with the fourth ventricle of the brain, and then you will have the spinal subarachnoid space around it, where there is arachnoid membrane, and then you have dura mater, and that is when you have a spinal vertebra around it, alright. So, that is the structure of the spinal cord, where you can see there are multiple nuclei within the ventral horn. These are anterior horn cells. I am sure you must have, all of you must know Stephen Hawking, who had these motor neuron disease and multiple sclerosis and other various neurological illnesses, whereas celebrities having these problems. So, this is where the problem begins. Now, they have this anterior horn cell being affected, where the cell starts to die, and they will have what is known as lower motor neuron paralysis. So, this is where the problem starts, where there is a grey matter and the cell starts dying, and then obviously the axonal bundles distal to it start degenerating. And what is outside the H is the white matter zone, where you have fiber bundles. These are dorsal funiculi, which will carry the touch sensation, will carry the vibration sensation. On the lateral side you have lateral spinothalamic tract, which will carry the pain sensation. So, outside this H is the multiple white fiber bundles, inside the H is the grey matter, where you have in the dorsal horn you have this laminar architecture. If you see, the neural bodies are organized in laminar architecture, whereas the nerve impulses which comes, which will terminate in a very specific layer, which is in the mathematical order, alright. So, the sensation from the thumb goes to very specific cord segment and synapse with the very specific lamellar architecture for layer 1 to layer 7 or 8. This entire layers are dealing with sensation, alright. These entire section is dealing with the motor system, which will have multiple interneurons. The impulses comes from one axon, synapse with the interneuron, and from the axons again there are multiple neural cells, and then it goes down. So, this will deal when we deal with the circuitry, but to know that there is grey matter, white matter, and there are various white fiber tracts suffices at this moment, alright. So, that is how the lamellar architecture is arranged. You can see there are various descending tracts and the ascending tracts. Ascending tracts takes the sensation from the periphery to the brain, descending tracts sends the signal from the brain to the end organs. That is very important to understand. So, and these tracts are only in the white matter. This is the grey matter zone which has cell bodies where its synapses, and from the second order neuron climbs up, then there is one more synapse relay junction happens, then the third order neuron goes to the brain there, alright. So, if your study involves a specific pathway, then you need to know where is the first order neuron, where is the second order neuron, where is the third order neuron. That would be the first order neuron, that will be the second order neuron, third order neuron going to the sensory system. If this is we are dealing with is a touch pathway. If it is a pain pathway, it goes here, ascends for a while, and then it crosses over, and then the second order neuron climbs, and then the third order goes to the brain. Here will be the thalamus. Thalamus is the major relay station, then it goes to the brain. So, we are not dealing with the actual nervous system structure, which is entirely a different session. So, grossly this is how the spinal cord is organized. A very organized structure, we can target very specific zone, and it produces very specific focal deficits in your rat behavior, alright. So, briefly I will introduce the spinal cord circuitry, that would comes to the end of the today's session. So, this is also very important, and as I said, it is like a classical electronic circuitry, you know. So, what is this is the dorsal root ganglia. I hope you all remember the gross anatomy of it. When you dissect it, this is how the neurons are organized. These are known as what is known as pseudo unipolar neurons, where one end of it actually is in the skin of the, let us say this is the forepaw. It carries the impulses and then relays with this, process the information, then it goes to the specific neuron in the layer 3 of the dorsal horn, then it synapses with the interneuron, then it sends the neurons right on top until the thalamus, alright. From thalamus then it goes to the brain, which is the third order, that forms the second order, this is the first order neuron. So, that is the entire circuitry for the touch sensation, if I were to say. So, similarly there are multiple neurons which comes from top. For example, if this is a motor cortex neuron, it directly descends to the interneuron in the spinal grey matter, and then it ends up in the alpha motor neuron, then it goes directly to the muscle belly, alright. So, this is the alpha, this is the interneuron, this is the descending corticospinal tract, which is known as pyramidal neuron. So, that is how the entire spinal cord circuitry, and this circuitry exists not only for motor and sensory, there is what is known as autonomic nervous system, and then there is a vestibular spinal tract, then there is reticulospinal tract, which all deals with the autonomic function, balancing function, and various other functions. So, there are n number of tracts and n number of circuitry, but this is the basic idea behind the basic structure of those simple circuits to begin with, just to sensitize as to how these circuitry are organized and how to target each of these structures in order to take your experiment forward. So, ultimately your surgical experiment depends on what is the objective that you are trying to address, and what is the question that what is the research question that you are trying to answer. Once that is formulated, then you really need to decide which section of the spinal cord are you trying to deal with, and then your surgical protocol starts getting designed. But then in next session we will try and look at various surgical protocols which forms the sort of foundation for various surgical experiments. Whichever spinal surgery you are planning, you have to go through those basic steps with little bit of improvisation here and there, and modification and customization as per your experiments. So, that covers the entire spinal anatomy and the anatomy of the spinal cord. In next session I will take you through the various steps that are needed for the exposure of the spinal cord, and then a brief introduction of various spinal experiments that are done in in the recent past, which are done in the recent past, and also the potential of future research project that we can discuss. Thank you all.